I should introduce myself. My name is Mark Baumgartner. I'm a senior scientist at the Hole Oceanographic Institution, and I'll say a little bit more about why, what my experience in this realm is. But just to go over what we're going to do today, I'm just going to give you basically a lecture on cetacean habitat modeling. I already shared most of the slides with you already. If you've had a chance to look at that, that's great. If not, that's fine. I'm going to try to not go over an hour. I shouldn't go over an hour, but it's going to be fairly high level, and it's going to be based heavily on my experience. And so it's not going to be the latest and greatest because I can't keep up with the latest and greatest. But in my view, what I'm going to talk about is essentially what's being used in the literature today, and with some modifications for other applications. So I only have an hour, so we're not going to touch on everything. But if we want to, as a group, dive into much more specific issues, I'm happy to facilitate that for a future little mini workshop. Okay. So after I talk, Hansen's going to talk about a project that he did for his PhD work to kind of put some meat on the bones for some of the things that I'm going to say, and then Catherine's going to talk about the work that she and Leslie are starting with a bunch of glider data from the New York Bight from both Stony Brook and Rutgers. Okay, so. Why me? Why am I doing this lecture? I've actually, and yeah, why is the glider acoustics guy giving this lecture? So I didn't start out as a glider acoustic guy. So for my master's work, I actually did habitat modeling is a bit grand, but tried to understand the habitat of donocetes in the Gulf of Mexico. And based on that experience, when I went to grad school, I realized I needed a lot more statistics to understand these tools that I and many other people were using. So I went to Oregon State for my PhD, and Oregon State has a fantastic ecological statistics program. So I actually got a PhD minor in statistics. I think I may have taken more statistics classes than I did oceanography classes. And then applied that for my PhD work, which was on North Atlantic right whales, trying to understand what are the oceanographic processes that influence the distribution and occurrence of right whales. Throughout the 2000s, I was involved with many other people in organizing a biennial workshop that was attached to the Society for Marine Mammalogy's biennial conference, specifically on marine mammal habitat modeling. And that went on for a number of years and culminated in 2013, where a small group of us, put together a special issue in endangered species research to say, okay, what have we learned over the last decade in habitat modeling? So this is something that I've thought about for a long time, and have opinions about, or a philosophy that is what I'm going to share with you over the next 50 minutes or so. Okay, so absolutely the most important thing you can do if you want to get into this work is to try to articulate why it is you want to build a model. So do you want to just do prediction only? Do you want to just estimate, say, density of animals and then extrapolate that to areas where we don't have a lot of observations, and you're going to use sort of ocean conditions to help in that extrapolation process? There's lots of studies that do that. And that's something that can be really useful to do. Or are you more interested about what oceanographic processes or behavioral processes influence the distribution and occurrence of your species, or density of your species? But yet you don't have a whole lot of information to go on. You just maybe you're studying a species that just there isn't a lot of known about them. And you're just trying to do sort of an exploratory study. And then there are those species where we actually have a lot of information about, and you're actually have used that information to come up with specific hypotheses about what drives distribution and occurrence. And in that case, you can use models sort of in a hypothesis testing mode, where your ability to make inference, you formulate the models in such a way that your ability to make inferences is very strong. And so you can actually do this hypothesis testing and believe, we'll talk about p-values a lot in this talk, you'd be able to believe the p-values and that they're genuinely telling you something about whether your hypothesis is right or not. Jessica Redfern wrote a great paper. I hope you all get a chance to read it. This is a long time ago. This was a product of a workshop a long time ago. For some reason, I feel like Joel might have convened this workshop. But what came out of it was Jessica Redfern led a paper describing sort of the state of the art in cetacean habitat modeling. And there's this figure in there that sort of helps me understand where you are on the habitat modeling spectrum. Do you know kind of nothing about a species? So that's where you have non-existent a priori knowledge, and maybe you're just going out to collect data to say, okay, to do something as simple as we found a bunch of these animals in this spot and we didn't find them in this other spot. So what's different from the standpoint of the ocean between those two spots? And maybe you're just going to use really simple descriptive statistics to do that kind of a study, just to sort of build up your ideas in sort of an exploratory way of figuring out what might be driving this species distribution. And maybe as you get to learn more, you get a little more comfortable about predicting the distribution of this species just based on the sort of environmental variables that fell out of this descriptive statistics study. And maybe that's as far as you want to go, but if you really want to understand how the ocean influences the distribution and occurrence of a species, you might start to get into hypothesis testing, where these hypotheses testing exercises are aimed at very specific oceanographic processes, very specific mechanisms. Something happens in the ocean, and this is how does it affect my species of interest, their distribution and occurrence. And so there's this continuum of knowledge that you can plug yourself into. So for North Atlantic right whales, for instance, we're on the really far side, on the right-hand side of this. For Kogia, we're maybe way on the left-hand side of this. And then there's different things that you might want to do with your models. And there's a bunch of different management applications. Maybe you want to do density estimation. That's certainly something that's very important task that National Marine Fisheries Service has to take on. Maybe you want to come up with ways, a habitat model that helps direct assets, aerial surveys, shipboard surveys, to some new area where you think maybe your species of interest might be, and that the aerial or shipboard survey will have a good chance of bumping into that species. And then finally, many of us are working on mitigation applications where you're trying to mitigate the impacts of industrial activities in the ocean. So it's really important that you understand why you want to build a model, because that's going to start to dictate choices that you'll make as you go along. There's a bazillion modeling frameworks that you could use. Generalized additive models are extremely popular, and in my mind are often poorly understood and overused, not necessarily the best tool for the objectives. And that's the point. There's nothing wrong with generalized additive models. It's just they should be applied to problems that are good for generalized additive models to address. There's also generalized linear models, such as logistic or Poisson regression, that I find to be handy. They're not good for all problems. Then there's sort of these, what I mentioned before as descriptive statistics. There's sort of ways to go about trying to tease out what is important, what what factors in your environmental space help to discriminate, for instance, between different species distributions? Canonical discriminant function analysis is really a handy way to go at that. Is that a model in the sense that a GAN is a model? Not really, but it's a tool in the toolbox, and it's definitely worth looking at, and sometimes can be more valuable, particularly when you're looking at how community composition changes with environmental variability. That can be, I have found that to be a handy tool. Same for a multivariate analysis of variance. And then there's a whole lot of other tools: occupancy modeling, Maxent. The statisticians on this call could probably rattle off 10 to 15 other things that could be used for this. But the point is that your choice of model really needs to be informed by why it is that you want to do this model and what it is you want to get out of it, what your research objectives are. Okay, so let's talk about what would go into your model. So what could your dependent variable be? Dependent variable meaning, what is it that I want to model? What is it that I'm interested in? What is it that has variation in it that I'm interested in explaining? One could be just simple presence and absence, looking at the probability of occurrence, for instance. And that's something that's very amenable to acoustic data, for instance. Not all species, we can't estimate the density of all species. Depending on how you talk to, they'll have different opinions on which species are good or bad at this, but we can at least say there's one or more of an animal there if we detect calls. So presence and absence is sort of a baseline thing that anyone can do with acoustics. Density, actually estimating the number of animals that are within detection range of your acoustic device, or on a ship track. A lot of what I'm going to talk about is equally valid for sighting surveys versus acoustic surveys. So maybe the a number of animals per square kilometer in your study area. Sightings per unit effort or detections per unit effort is another way. Instead of saying per kilometer, it's per square kilometer. It's sightings or detections per kilometer along your transect. And then call rate, which for acoustic studies we have available to us, just how many calls happened in this kilometer or in this hour of data collection. That's a funny one because it incorporates both behavior and distribution, spatial distribution. So it's one to be careful about, or at least thoughtful about, if you're going to use that as your dependent variable. Again, this choice of which one you're interested in really goes to why are you doing this modeling exercise in the first place, and what do you want to get out of it? I suspect I could put this choice is informed by purpose of modeling on every single slide, so I think I'm going to stop doing that. So how is it that you should treat your dependent data? So, okay, I'm going to do presence, absence, or I'm going to do density. So I'm going to stick that into the model. Do I just stick it in and go? Well, that depends. Depends on your purposes, but it may be helpful to aggregate or bin spatially or temporally based on something called autocorrelation. So I'm going to talk for a little bit about this idea of autocorrelation. So what autocorrelation means is that and we'll get into this in more detail in a second, basically means that if I make a measurement now, how long into the future will I get a measurement that's completely unrelated to the measurement I just made? We'll talk about temperature in a second, air temperature. So if I take the temperature now and I take it in an hour, that temperature is going to be pretty similar to the temperature that I just took. And if I take it in 2 hours, it'll still be pretty similar to the temperature I just took. And if you keep going out in time, eventually you'll get to a time scale in which the temperature that you measure at that distant time has nothing to do with the temperature that I take now. And so that time scale is something that we're going to want to measure, because that's going to be important. Why is it important? In any model, in most model applications, you want your dependent observations to be independent from one another. If they're not independent of one another, you run into this problem called pseudo-replication. You basically fooled yourself into believing that you have more observations than you actually do, because your observations are not actually independent of one another. And so we're going to use this autocorrelation sort of methodology or idea to guide over what minimum temporal or spatial scales you should be aggregating your data. If you don't do this, and you're interested in interpreting the p-values that come out of whatever model you're using, if you don't deal with this autocorrelation issue, you have this pseudo-replication problem. Your p-values are just not going to be accurate. They're going to be smaller than they ought to be, and you're going to get fooled by this. So if you've had a chance to run, to do this kind of modeling, and you get a model where every single variable has a p-value that's like 0.0001, that's something where you'd be like, okay, wait a second, why is that the case? And you may, if you back up, you might find out I don't actually have my observations aren't independent of one another. And so I'm getting these spurious associations because the p-values are just not believable. So in some cases, this is fine. If you're just predicting or you're doing exploration, data exploration, this pseudo-replication is something certainly to be aware of, but it's not a deal breaker. As long as you're wary, you sort of approach your p-values with some caution, it's fine, because you're just doing an exploration. But if you're doing hypothesis testing where you're depending on those p-values to be accurate, to be telling you something about the true sort of associations in your data, i.e. you're trying to make inference, then this idea of pseudo-replication is absolutely critical. So I'll just very quickly go through autocorrelation for a very simple time series like temperature. So I'm just measuring temperature, say, in this room over time. So autocorrelation is just, I'm going to take the time series of temperature and I'm just going to shift it by a little bit of time. And then I'm just going to do a straight up correlation. I'll get a correlation coefficient and I'm going to plot that on this graph on the bottom. And then I'm going to shift that original time series one more time step, and I'll plot the correlation between the original time series and the shifted time series. And I'll just keep going, shifting, taking correlation coefficient, shifting, taking correlation coefficient, and so on. Eventually, I'll do that, and I'll come up with an autocorrelation sort of time series where it's based on the time scale. And what's important here is where this autocorrelation function crosses zero. Where it does, that means the measurement that I made out in time compared to the one I make now, if I make it at this decorrelation time scale, that measurement should have nothing to do with the measurement I'm making now. That means if I chunk all of my data at that time scale, I'll have independent observations in this case, temperature time series. And again, air temperature is a great way to think about this. I can measure air temperature 500 times a day and do that for a month. Does that mean I have 15,000 observations of temperature that are useful to put into a model? No, because any temperature measurement that's adjacent to one another in space are not independent whatsoever. I have to do this, find this decorrelation time scale, and then bin my data according to that decorrelation time scale. And that's how much data I really have to be able to say something meaningful about temperature trend or whatever it is I'm interested in. Okay, so let's say you had some glider data. You've got a glider track, and you've got some, in this case, let's just say it's, I detected a call of a particular species at each one of the stars here. Many of you who use sort of acoustic data either through time or space know that calls come in bouts, they come clustered. And so if you're just chunking your data up into every single call, like every minute by minute, your observations are going to be highly correlated with one another. And so you need to figure out what's the time scale that I could use where my observations are independent. And that's what this autocorrelation analysis does. It depends how you come up with this autocorrelation sort of analysis, depends on the type of dependent data you have. So I'm not going to go into that, but the concept is the same. You want to find out what this decorrelation spatial scale is so that you're dealing with cluster detections and you're ensuring independent observations. Last thing I hear often is like, I'm really worried about autocorrelation in my independent variables, my explanatory variables in my model. And that's not something that you have to worry about at all. Autocorrelation in your dependent variable is important because of pseudoreplication. Autocorrelation in your independent variables is not a problem at all. There are other things that we need to think about with your independent variables that I'll talk about in just a second, but autocorrelation is not one of them. Okay, well, let me just pause there. Does anyone have any questions on any of that? Shout out, because I'm not sure I can see hands. Okay, I'll keep going then. Yeah, go ahead, Andrea.
So why is the autocorrelation in the independent variable not a problem at all? I was wondering. I'm still trying to grasp that.
Let's say your dependent variable has sort of a decorrelation spatial scale of like, I don't know, I'll just make it 5 kilometers. But you have an independent variable that doesn't vary a whole lot. It varies over maybe spatial scales of 20 kilometers. Well, in your model, every 4 observations of your dependent variable, there'll be no change in your independent variable. That's fine. That doesn't affect the p-value, for instance. That's just you have a variable that doesn't vary very much spatially, and that's fine. Your analysis will sort of, if that variable is in fact important, the model will bear that out, and you, as long as you've dealt with this autocorrelation in the dependent variable, your p-values would be completely unaffected by the whatever spatial scale your independent variables vary over.
Thanks.
All right, let's get on to the independent variables then. So what environmental variables should you use? So this is a kind of a funny laundry list, sort of from the easiest to the not hardest, but most targeted. So let's say you don't know anything about the species that you're modeling, and so you might just grab variables that you have access to. You don't have any a priori hypotheses about what drives this animal's distribution. In fact, you know nothing about what drives this animal's distribution, and you just happen to have access to, I don't know, water depth. Great. You could certainly do that. You could just grab whatever's available to you. You could also look to the literature and say, well, what other variables are used in modeling studies? And there's a couple ones that you'll find over and over and over again. Sea surface temperature, surface chlorophyll, water depth and topography are always in these types of models, and for good reason, often. So that's great. So those are also variables that are readily available, and I think that's why we see them in so many modeling studies, is because sea surface temperature is widely available from remotely sensed sources as well as surface chlorophyll. If you have some knowledge about the environment in which you're doing your modeling, then might be useful to grab variables that represent sort of the dominant oceanographic processes in your study area. And this requires you to either you yourself or to be collaborating with someone that really understands the ocean in your study area well. So what is it that's important? What are sort of the dominant oceanographic processes affecting spatial and temporal variability in the ocean? If the species that you're looking at there is a lot of information about that species, even though it might not be about that species' spatial distribution, but it might be something that affects their behavior or their temporal occurrence, then you might go and look to grab those kinds of variables and put them into your model to see if they affect the spatial distribution of your species. And finally, for those species where you've got a lot of information and you've got a really good understanding of the ocean, you might start to be able to hypothesize what oceanographic processes are actually governing the distribution of your species, formulate variables that represent those processes, and then include those in a model where you're actually doing hypothesis testing to see, is this process really having an influence on my species distribution? So there's lots of different reasons why you might use these variables. What I'm going to stress here on out is just be really thoughtful about it. Just ask yourself, why is it that I'm including this variable in my model? What is it going to tell me? And how will I interpret it if it comes out the back end of the model as being significant? So some of you know one of my hobbies is brewing beer. And if you've ever cooked, it's the same idea. You have to be very intentional about the ingredients that you put into beer or food. You don't just go into your pantry and just grab whatever and throw it in. If you do, it's probably not going to taste very good. And the idea is kind of similar in modeling. You want to grab environmental variables and put them into your model in a very thoughtful and structured way. And that helps you actually interpret the model at the results of the model at the end. Okay, so for that endangered species research theme section that we did, we wrote a paper at the end basically saying, where are we going with habitat modeling in our community? And this was a figure pulled from that. And it was designed to sort of remind us of all the things that would influence the spatial distribution of our species. And so, environmental factors is just sort of one thing. And it's even amongst environmental factors, there are a number of different things that can influence spatial distribution. It's not just how hot or cold the water is. It's how much prey is there? What's the concentration, production, availability of that prey? What's the prey's behavior and life history? That has an awful lot to do with spatial distribution of predators. But then there's all these behavioral things like foraging adaptations, reproduction, and so on. So again, think about these things and what is it that might be influencing the spatial distribution of your species. Okay, with glider data, what's nice about gliders is you have this opportunity on a platform that profiles through the water column to measure a whole bunch of stuff. Not everything, right? It's a small platform. It's very sort of payload limited, but just from temperature and salinity profiles alone, you can derive a whole lot of things. And so here is where having an understanding of the local oceanographic conditions and processes in your study area is important. If you want to capture what's going on sort of environmentally in your study area, you really have to have some understanding of the oceanography of your region. And then you can start to pull out variables from those temperature and salinity profiles that may be meaningful in your area that may or may not be driving the distribution of your species. Here's just a couple. So surface temperature and salinity is very commonly used. Unfortunately, surface temperature and salinity are not conservative, meaning a large part of the variability in surface temperature and salinity comes from factors that are driving the atmosphere, not necessarily the ocean. Some of the variability in surface temperature and salinity has to do with the ocean, and that varies from place to place. In a place like the Gulf of Maine, the surface temperature and salinity is heavily influenced by the atmosphere. Someplace like in the Gulf Stream or the slope seas, where you have these mesoscale features like warm core eddies or jets, sea surface temperature can tell you a lot about the structure and of the ocean, and could be very, very useful. And so it's just, if you just have to know or consult with an oceanographer about what's important or not. In the Gulf of Maine, for instance, in a relatively on the continental shelf, bottom temperature and salinity sometimes is a much better indicator of what is sort of going on in your environment than surface temperature and salinity, because bottom temperature and salinity don't change very much. They only change with oceanographic processes, specifically advection and mixing. Taking a look at clines, at gradients, thermal, halo, or picnic lines, that's temperature, salinity, or density, can be very helpful, depending on the system, to define something like the surface mixed layer depth, or in the case, the next thing, the bottom mixed layer depth, which is something that we found to be important for determining North Atlantic right whale distribution. And then you might look at the depth of a specific temperature, salinity, or density value. So those can be really handy. I've certainly, in my experience, have used those to look at mesoscale features. So if you're off the coast of California, for instance, you're doing transects, the depth of a particular isotherm, maybe the 10-degree isotherm, can tell you a lot about whether you're in or out of a cold core eddy. There's other things that are pretty readily available on gliders. Chlorophyll concentration derived from fluorometer. This is something to be both happy and cautious about. It's nice to have the data, but you have to realize that it varies substantially temporally as well as spatially. And this will become a theme for the rest of my talk. Teasing apart spatial and temporal variability is a tough game with glider data. And chlorophyll is a great example of that. Chlorophyll can vary dielly as well as over seasonal timescales. Turbidity is pretty easy to get. Oxygen concentration is pretty easy to measure. And there's folks like Kim Davies on this call and Grace Saba that are looking at acoustic backscatter or even zooplankton concentration, say from a shadow graph. So there's lots of things that the gliders can lots of data that they can provide you. It's your responsibility to make that data into something very useful to actually say something about the ocean environment that your animals are experiencing, and therefore say something about what oceanographic processes they prefer or not. Okay, it might be that you've got this great set of environmental variables. Great. So your first instinct is just throw them into a model and let's see what comes out. Be careful about that. If you're interested in what drives spatial variability, then you want to include variables that account for temporal variability in the dependent variable. And this is quite critical for gliders because, as we'll talk about shortly, you can collect a lot of glider data, but most of what you're going to use that data for is to tease apart this temporal versus spatial variability. And you're going to have to add, you may have to add new explanatory variables or independent variables that try to capture some of the temporal variability in your dependent variable. I'll give you an example of that in just a second. There also may be times when you know enough about a species that you absolutely know that there are variables that have an influence on the distribution of that species. A great example of this is if you're doing glider transects that go from the continental shelf over the continental slope. You have this huge variation in water depth. And cetaceans tend to stick to particular water depths, particular depth strata. So there's a continental shelf community There's an upper slope community, and then there might be a lower slope and abyssal plain community. Well, if you know that already, then when you formulate your model, you should probably put water depth as one of your variables, because you know a priori that that will have an influence on the distribution of your species. So you want to sort of account for that right up front, and then see what other variable, and then your model is sort of focused on what other variables are important to my species distribution above and beyond depth. And then finally, there's these nuisance variables. They're variables that cause variability, and variables that cause variability in your dependent variable that you're not really interested in. And a great example of this is if you've ever worked with sighting survey data. If you model sighting survey data, you always put in something like Beaufort sea state. And that's to try to capture the detectability of the species that you're interested in. You don't care about Beaufort sea state, and the animals probably don't care about Beaufort sea state, but Beaufort sea state is absolutely affecting how well you're able to detect that species. And so in acoustics, you might want to include things like background noise, calling rates. I don't know how you would incorporate that, but it's something to think about. Okay, so how should I treat my independent data? So there's a couple things to think about. One is to use, certainly you're going to choose variables of interest derived from your data, meaning you're going to process your data, and you're not just going to stick in every single temperature measurement that you took from the sea surface to the sea floor for, the 6 years of data that you have. You're going to have to derive some variables that are meaningful, that you think are meaningful, either in describing the oceanographic conditions of your study area, or you think they're meaningful to the species that you're studying. It's important to consider using temporal and spatial anomalies when appropriate. And I'm going to give an example of this in just a second. But sometimes just sticking in temperature it may turn out that the species that you're interested in is not responding to temperature per se, but basically temperature relative to what's available to it at that time. And so, meaning most cetaceans are not constrained at all by temperature, right? They're warm-blooded. They can go where it's really, really cold. I mean, right whales go in the summertime, they dive down to where it's 4 or 5 degrees C, and then they come to the surface where it's like 20 degrees C. Like they can handle those temperature ranges, no problem at all. Cold-blooded species like fish are much more temperature constrained. And so this idea of using temperature sometimes crosses over from fish to mammals in a bizarre way. But it may be that temperature does influence, but in a way that I'll describe in a second that's quite different than what you would expect, in which case a temporal or spatial anomaly might be appropriate. One of the things to watch out for with your environmental data is if you have a bunch of environmental variables that are correlated with one another. That's called multicollinearity, and it's something that most modeling approaches are going to have trouble teasing apart. If your goal is to have a model tell you what's the most important environmental variable here, and 3 of the most important ones are all correlated with one another, the model's not really going to figure that out for you. And quite honestly, you're not going to figure that out either, because they're correlated with one another. And so that leads you to be like, okay, why are they correlated? It may be that there's some oceanographic process that's leading to those 3 things being correlated to one another. So what is that? That might be something to look into. So the correlation structure of your environmental data can be interesting in and of itself, but it's something you want to sort of resolve before you just throw all this stuff into a model. One nice way or clever way, although not always productive, is to figure out what are the dominant modes of variability in my study area. And there are tools to do that from the physical oceanographic community. One is principal components analysis. You could take all your environmental variables, do a principal components analysis, and do your model based on those, the principal components that come out of that analysis. You're assured that multicollinearity is gone from your environmental data if you use those principal components. But you may or may not be taking a step towards interpretability or not. So it's something to be aware of as a tool. In some cases, it can be a very, very useful tool. But again, it's really important to be thoughtful about your choice of environmental variables and then how you treat those variables in your analysis. So let me give you a quick example of this. And I'm not saying this is how you should treat your data. It's one way to think about treating your data. That's sort of one permutation that you can use or test or and to think about, again, to be thoughtful. So let's just say you did a survey. It doesn't matter if you did it with a glider, did it with sighting surveys, or I don't know, any other detection method. And so on the left, you have a survey area that has mostly cold water, and then in one area that's much warmer, it's a little bit warmer water. And you tend to see your species of interest, which are the stars, sort of clustered in the colder water. Then the next year you go out and you do the exact same survey in the exact same place. And this time, things have shifted. The background, the vast majority of the area has even warmer water, but now all your species, all your sightings or detections are in this cluster of even warmer water. So in this case, the spatial pattern that's true is that this species has a preference for the warmest water that's available to it in the study region. Okay. Clearly, there's interannual variability in water temperature. In year 1, it's colder. In year 2, it's warmer. We need to be able to recognize this and deal with it. So if you just took this data and threw it into, say, a generalized additive model, you might come up with something like this. So I'm hoping you can see my cursor. Yeah. Can anyone see my cursor?
Yes, we can see it.
Great. Thank you. So for year 1, you have low occurrence in 8-degree water, high occurrence in 10-degree water, low occurrence in year 2 in 12-degree water, and high occurrence in 14-degree water. Okay, so that's the relationship with temperature. And you can model that, and from modeling that, let's say it doesn't matter. So let's say we had a logistic regression. So occurrence, the probability of occurrence is related to temperature, T, in some way, using the parameters beta naught and beta 1. So in this case, if you just throw the temperature as it is into the model and you run the model, you'll come up basically with no relationship between your species occurrence and temperature. When in fact, that's not the case. We said that the truth is that this animal has a preference for the warmer waters that are available to it. And so if you want to sort of test that hypothesis, then one way to do that is to use a temporal anomaly of temperature as the independent variable to account for this interannual variability. What I mean by temporal anomaly, it's the temperature of the water minus the annual average over the entire study area. If you do that, you come up with the correct interpretation of what the spatial pattern is. That is, in a year where in space that have a low temperature relative to that year's average temperature, occurrence will be low, and in areas where the temperature is high relative to that year's temperature, occurrence will be high. And the only thing we've changed in this model is to take the average annual the annual average temperature and subtract it from the actual measured temperature. And then we get the truth. So this is all I'm trying to say here is that if you just take your variables and throw them into the model without thinking about what's possible or what the possible relationships are between your species distribution and the environmental variable, then you could be led astray. I'm not saying that this is the case. This is one possibility that you might test for in your data. It may be that your species doesn't have a preference for warmer waters available in your study area, that they have a rigid temperature preference, and that's fine. You can formulate a model to test that idea out as well. So this is a case where there's just interannual variability in the environmental variable. Well, what happens if there's interannual variability in the occurrence of your species as well? Same exact example. There's year-to-year interannual variability in temperature, but now there's year-to-year interannual variability in occurrence. For some reason, the occurrence of your species has gone down in year 2. And so you still have this preference for warm waters available in the study area, but you have interannual variability in both water temperature and occurrence. So this is what this would look like. So in year 1, 8 degree is low occurrence, high occurrence in 10 degree. In year 2, it's even lower because you have lower occurrence in year 2, much lower in the 12 degree and modest in 14 degree. Well, how do you deal with that? In this case, your view, if you don't account for the temporal variability, your view is exactly opposite what the truth is. This animal has a preference for warmer water, and your model is saying that there's actually occurrence goes down with warmer water. So you're going to need to use temporal anomaly of temperature as we did before, but now you're going to include a year term to account for interannual variability in occurrence. And when you do that, you get the correct spatial pattern after you've accounted for interannual variability in temperature and interannual variability in occurrence. And that equation that I just wrote up there is how you would formulate this model using like a logistic regression, Poisson regression, whatever you want to think. But a good way to think about this is this way: occurrence minus your annual sort of average occurrence value is related to temperature minus its annual average temperature. So basically, you're making an anomaly for both your dependent and your independent data. This is kind of, as Hansen, you'll see in the next talk, Hansen and I went around and around and around, and this teasing apart this spatial and temporal variability is a real challenge with particularly with glider data because they move so slowly. Okay, let me get finished here. Only a few more slides left. So it's important to keep in mind how much data you do and don't have. And there are some rules of thumb. I'm sure the statisticians on this call could give us better rules of thumb. But for generalized linear models, the ones I use the most, a very rough rule of thumb is to have at least 10 observations per variable used in the model. If you only have sort of 50 observations, you're not going to be able to throw 30 environmental variables into your model. You're just going to overfit. It's going to be a disaster. So you need to be mindful of this. This also goes into your choice of what modeling approach you use. Generalized additive models need more data than even a generalized linear model because you're fitting more parameters to allow your curves, your lines to basically curve. You need more observations. So something that's quite important to think about. And then oftentimes people ask, like, what's the right spatial and temporal scale? And so we talked about space-time autocorrelation in the dependent variable, which drives the minimum spatial or temporal scale you can look at. The maximum spatial or temporal scale that you can look at is really governed by how much data you have. And you'll be shocked to learn how little data you have when you do all this autocorrelation stuff. It's often surprising. So What's the right spatial temporal scale? You have bounds on that, but you're free to choose within that. And then maybe you start to think about, well, what are the temporal and spatial scales of the oceanographic processes that I'm interested in? And if those fall within those bounds, maybe you should focus on those spatial and temporal scales. Okay, just very quickly, a lot of people, one way to do this is to throw everything into a GAM and basically step back and let the GAM pick all the things that are important in your data, all the important variables, and then to give you statistics to say, okay, how important are those variables? And what these modeling processes are doing is some sort of selection. It runs a whole bunch of models and says, okay, which ones fit the best? If I take this variable out and I put this variable in and I take that one out again and put another one in, that's called forward or backward variable selection. And one of the things you have to think about when doing that is that has an effect on the interpretability of p-values. If you let the machine do all the model selection for you, you ought to be a little careful about using p-values that come out of that, because there's been a lot of sort of, you've cooked the books a little bit to get the best p-values possible. And then if you interpret those p-values as if you had just run one model and chose those variables a priori, you wouldn't have gotten those same p-values. So you have to be really careful. So model selection sometimes is not really a good technique for hypothesis testing. It's great for exploration and prediction, but not so much for hypothesis testing. And something to often think about is whether the best fitting model is really a reflection of the truth, or is it just the model that best fits the data? And if your model is challenged with independent data, would it do so well? Another thing that's related to this is managing multiple comparisons. You might end up running a whole bunch of models to figure out what environmental variables are important. But the more models you try, the better chance that a non-significant model will appear as significant. And so you need to be aware of that, and you need to sort of figure out a way to deal with that. And one way, again, Hansen will talk more about this, is to consider something as simple as a Bonferroni adjustment, which is just an adjustment to the alpha level at which you consider a p-value significant. The final thing I think is to think, as you're formulating your model, think about how is it that I'm going to interpret this model, even before you get any results. So a lot of people like interaction terms. They like to throw interaction terms in because they're like, well, I don't know if this is a linear relationship or something else. Many times interaction terms can be difficult to interpret unless you use them quite thoughtfully. And so there's nothing wrong with using them, but do so in a way that helps you figure out on the back end, okay, here I got all my results. What do they actually mean? Having a whole bunch of interaction terms can often make the results very difficult to interpret. It's the technique du jour of generalized additive models. I've seen many papers and presentations where the end result is like 8 GAM curves, and the person that's presenting it is like, well, here are the results, and here's they usually don't say what it means, because it's very difficult to interpret 8 GAM curves, 8 different associations between your dependent variable and these 8 different environmental variables, and say something meaningful about what's actually going on. So just be intentional about the variables that you use and what you would expect to get out of a GAM curve, or a set of GAM curves. And then finally, I keep hammering this, be careful about trusting p-values. Have you formulated your model in a way where you can be confident that the p-values are telling you something meaningful, or are those p-values a little squirrely because of pseudo-replication or model selection, your model selection process? Okay. So you want to go out and use your gliders, and you want to do some sort of habitat modeling. How should you design a survey? Because of this, teasing apart spatial and temporal variability is so challenging. It really helps to visit the same areas at the same time of year, so you can actually quantify interannual variability. If you go to a different place at a different time of the year and you never visit that place at the same time of the year again, you've got basically essentially a sample size of 1 for that area and time, and it makes it really hard to tease apart temporal variability from spatial variability. The slow speed of gliders makes collecting sufficient data for modeling difficult, given the detection ranges and spatial autocorrelation for many species. Again, I think you'd be surprised at how little data you have after many, many years of collecting data. So many years will likely be needed. And if you survey in different areas at different times, this becomes difficult. So again, it goes back, part of this goes back to the survey design is what environmental variables, what oceanographic processes are you really interested in looking at, or do you think are driving the distribution of your animal? And design your survey around sampling those sort of oceanographic areas or provinces. The last thing, this is the last thing I'll say, is that be careful with glider data. The data are not synoptic, meaning it's not a snapshot. This isn't a satellite, snapshot at one place in time, one moment in time. So a good example is I have a glider out right now in the Gulf of Maine. It's doing a survey along the northern edge of Georges Bank, and it's actually right now on its way back. And I'll pick it up off of Chatham, Massachusetts at the end of July. But look at the fin whale in the upper right. So red means detected, yellow means possibly detected. You note that just above Georges Bank, we had fin whales galore on the way out. But now on the way back, we're surveying just to the north of where we were surveying before, and all of a sudden fin whales are gone. Well, what happened? Did I just design the survey perfectly to bisect this area to the south where there's tons and tons of fin whales and this area to the north where there are no fin whales? Probably not. It's probably that there's some temporal variability going on here. When I did the survey just north of Georges Bank, the southern part of this survey was at one time of year, was like April, May, and now I'm doing the return survey to the north of the southern part during June and July, a different season, from even going from spring into summer. So if I just showed you this map, you'd be like, oh wow, there's fin whales galore just north of Georges Bank, but not in the central Gulf of Maine. And I would say, well, I'd be really careful about that because they were sampled at different times of the year. Okay, that's all I had. I'm done.